So today I'm going to talk about tumors. What are tumors, how many kinds of tumors there are, what they can do to the body, and more specifically, what does calculus in tumor growth, what does calculus have to do with tumor when monitoring tumor growth? So my project will be on how calculus is applied in monitoring tumor growth. So calculus is used when a tumor is found by measuring the volume of the tumor as well as using calculus to figure out the growth rate at which the tumor is growing. I'll be going deeper into detail on what that looks like and how it's used. I'm sure a lot of people have heard about the word tumor, whether it be real life, TV, or in school classes. Anyone can look up tumor and learn that in a simple definition, it's a mass or lump in the tissue that may resemble swelling. Well, more specifically, according to an article titled, What are the Different Types of Tumors? Medically reviewed by Christina Chun, MPH, written by Yvette Brazier, August 21st, 2019, tumor is defined as and with normal mass of tissue that results when cells divide more than they should or do not die when they should. When people hear the word tumor, their brain usually shoots to the word cancer, and while some tumors can be cancerous, there are tumors that are not. And that's when the different types of tumors come into play. There are 3 different types of tumors: benign, premalignant, and malignant. Now, benign tumors are not cancerous. They cannot spread or grow, but if they do so very slowly, and once a doctor removes them, they are not likely to return. Malignant tumors are cancerous. The cells can grow and spread to other parts of the body, which can result in death. And now premalignant tumors are tumors that they don't have cancerous cells yet, but they do have the potential to become malignant and or cancerous. It's not always clear how a tumor will react in the future. Some benign tumors can become premalignant, then malignant, or cancerous. And for that reason, that's why it's best to monitor any growth. And over here, the picture, it will tell you more about benign and malignant. So benign is non-cancerous, it's capsulated, non-invasive, slow-growing, does not metastasize or spread to other parts of the body, and the cells are normal. Now, malignant tumors are cancerous, non-capsulated, fast-growing, metastasized, spread to other parts of the body, and cells have a large dark nuclei which may have an abnormal shape. All right, now on to how math is used and its importance in the tumor growth. According to a Western Kentucky University Sevgi Sengul's thesis titled Discrete Fractional Calculus and its Applications to Tumor Growth, May 2010, states that mathematical modeling of tumors began in the early 1950s after scientists discovered that the initiation of cancerous growth could only occur after multiple successive mutations in a single cell's DNA. Now, math plays a very important role when monitoring tumors and its growth, and we monitor by size, volume, and time. These are the 3 mathematical equations used. They are exponential, logistic, and sigmoidal. And here we have, for this symbol here is lambda, which defines the growth rate. And this symbol here, theta, defines the carrying capacity. So the basis of any mathematical model used to study cancer is a model of tumor growth. A number of ordinary differential equation models, or ODEs, have been proposed to represent tumor growth and are regularly used to make predictions about the efficiency of cancer growth. A lot of researchers have attempted to find the best ODE growth model. When it comes to the data sets of a tumor growth, there are 7 ordinary differential equations, or ODE, models of tumor growth: exponential, Mendelssohn, logistic, linear, surface, Gompertz, and Bertalanffy that have been proposed. But altogether, the results of all of those have been inconclusive, and they did come to the conclusion that tumor growth is best described by the Gompertz equation. Now, according to research paper titled Interpolating and Gompertz Function Approach in Tumor Growth Analysis, written in 2018, Ogunrinde RB and Ayinde SO state that Benjamin Gompertz originally created the Gompertz model in 1825 in order to explain human mortality curves. In 1939, the model became a generalization of the logistic model with a sigmoidal curve that is asymmetrical with the point of inflection. The curve was eventually applied to model growth and size in entire organisms and more recently was shown to provide the best fits for breast and lung cancer growth. The Gompertz difference equation describes the growth models, and these models can be studied on the basis of the parameters in the recursive formulation of the Gompertz law of growth. So here is shown the Gompertz equation. And here it's where n equals n to t is the population of tumor cells, where r is the constant growth of cells that are where r is greater than 0. K is the carrying capacity of the tumor. The maximum size that it can achieve with the available nutrients is based on the findings he generated with the following facts: that the carrying capacity K of a tumor will be intimately related with the quantity of tumor cells. The Gompertzian analysis of altercations of tumor growth patterns. So applying fractional Gompertz model results to human renal cell carcinoma, which is a malignant tumor of the cells that cover the line of the kidney in nude mice. The nude mice inhibit the immune system due to a regularly reduced number of T cells, and the parameters for growth control are taken and shown here in this table or graph. So here in this figure, 5.a, it shows the control growth with parameters a equals 2, b equals 0.85, and c equals 1. We draw the control growth with different a values. First observation in here is when a is to 1. Fractional mode converges to the continuous model as expected. The convergence is clear for low values of t in figure 1, or figure 5.1, sorry. So a values other than 1 behave like the growth declaration. Since the parameter b represents the exponential growth declaration, next we change b to observe the growth behavior. So in figure 5.2, we see that if we take b equals 0.9, we observe simulation for each a value. It could be seen that in this graph, curves y and z behave similarly. Y is the curve with b equals 0.85 and a equals 1. And z is the curve with 0.9 and a equals 0.83. This shows that we can observe different behaviors by changing the parameters a in discrete fractional calculus. So if we set b to 7, which is less than the control growth, we observe inhibition for each a value. So based on the results above, we also observe that the growth behavior shows significant variations with respect to a using fractional calculus for curve fitting. We could get more accurate results from data analysis since we have more fitting parameter a in addition to the model parameters. To support this insight, we compare our model for a different a values with a continuous model using real data. So here we have the derivatives of the ordinary differential equation models, or ODE. The derivatives of the interpolating function are required to coincide with the differential equation, as well as its first, second, and third derivatives with respect to x at x equals xn. So here we have our a1, a2, and a3, which are our first, second, and third derivatives with respect to the x at x equals xn. So to wrap up our conclusions, as increasing variety of mathematical models have made its way into cancer research over the past decades, especially in tumor growth problem, little is done on using numerical integration to solve this problem. In this research work, we illustrated how quantitative models in terms of interpolating function can be developed and analyzed into numerical integration and used experimental data to show how it can stimulate complex biological process in tumor growth. So the logistic model or equation used in this analysis, as well as Gompertz equation, can be modified as it's being done by many analysis. So we learned what are tumors, how math is important in monitoring tumors, how math is used when equations play a role, how math is used in which equations play a role, insight to the graphs and derivatives on the calculus being used, and the Gompertz equation. So once again, tumors we know now are an abnormal mass of tissue that result when cells divide more than they should or do not die when they should, resulting in a clump of cells that, when cancerous, spread to other parts of the body, resulting in more cancerous cells, which could result in death, and benign tumors, which are cells that are not cancerous and could be removed easily and do not come back. So for the math, it's important because we need to know how fast they're growing and how big they're getting in that time. And that's when the Gompertz equation came into play. And that's when they found that's what was best when trying to monitor the volume and size and the time.